Good afternoon. After a long talk by professor, and it was like so. Before starting off with something called machine learning in mapping the connectivity of the human brain, so from the first day we are hearing that machine learning has been a crux of this neuroscience. So how far is that it has gone? Just to give you an overview of that. So this is the kind of white paper by Signify Research, uh, where they had 7 predictions, and all these predictions says that machine learning, the first prediction you see that it will be the machine learning in diagnostic imaging will top 2 billion by 2023, although it will improve radiology by patient experience at every step. And to sum up with that, we also have kind of the US FDA, which has actually cleared an AI algorithm which can provide clinical decision support. It is kind of a very big achievement for the AI community that the US regulation systems have actually passed an AI algorithm. Uh, Although we are seeing a lot of improvement in the machine learning algorithms, there are a lot of new use cases coming up. And to be very true that we don't have much of we do have information, but we don't have much of label data to really work on the machine learning algorithms. I have been working on machine learning since my PhD, but while going into neuroscience, we are really having kind of a problem having the kind of label data to uh, really have annotations on different kinds of algorithms to have it supervised. Although unsupervised algorithms perform to some extent, but to reach a kind of accuracy where the health system really will believe us, we need some supervised data. So this is kind of new use cases which come up every day, and all humans are not the same. The diseases and the different kinds of trends are always not the same, which doctor or a physicist can easily identify. So these new use cases come up to the machine learning algorithms, and we have to really kind of train our algorithm or tune our algorithm to those tasks. So new use cases come up, and it will be coming up day by day, because with the advent of new viruses, new kind of diagnostics, it will always come up. And also, this is kind of very interesting. So the prediction 4, medical imaging AI will transform from academic research to commercialization, which is already started in US. What I look at, and we can see there are several companies which actually kind of try to use AI for different kinds of doctor assistance, as well as it will benefit different kind of medical imaging. So I'll talk of not a kind of medical imaging, but a kind of brain imaging. So in machine learning, where we'll try to it is a mouse brain, so we'll try to look at how the connectivity of the different neurons in the brains look like. There are also like the prediction 6 also will use the different data hubs to unlock the value to the data. There are kind of AWS servers which are now really dedicated for health engineering, and even kind of having medical image processing algorithms inbuilt on them. There are specialized TPUs being developed by Google, even to support this kind of machine learning tasks. So there will be high development in IT infrastructure, and it will also help to bring up medical imaging AI systems to a practicality and even to commercialization. These are a few of the achievements. You can see that in a CWRU in 2017, they actually had found out that they have identified the invasive form of breast cancer from pathology images, although to 100% accuracy. So this study is already available on the website, and you can also find that at Stanford School of Medicine, we can see there is a machine learning algorithm which accurately differentiates between two types of lung cancers. Although I am not a kind of medical person, I am kind of a computer scientist. I don't know much of what kinds of the lung cancers, how many kinds and how many types are there, but each can distinguish between two types of lung cancers, and that has been, the accuracy has been rising, and it is almost above 99%. So what we talk about here is like, we really need to look at machine learning algorithms having a very high accuracy. To talk of accuracy, it needs to have a very high recall. So the main drawback which we are facing here is like, we are not an EM community. We are a community, EM like the electron microscopic community, which has a huge data annotation centers, data annotation teams which work together, and they are like kind of a mature community in the field of medical imaging. But still, the NIH Brain Initiative, which we actually are a part of, so we really are working on kind of getting this image processing network or the machine learning network to work really to a kind of high accuracy to really achieve that goal. So the recent April 2019 issue talks about those initiatives. You can really read through this current opinion in neurobiology, where Jonathan Pillow, this guy, and Manish Sahani, they are the Siemens collaboration. They are partners in Siemens collaboration on the global brain. They talk about this initiative and their goals. So the Brain Initiative Cell Sensors Network is like, there are 4 groups which has their kind of expected outcomes to be kind of achieved by, they targeted by 2025. So I actually listed down those outcomes. So they tried to gain a knowledge on the different cell types, on the 3-dimensional organization of the logic in the brain, like the connectivity. They have to have a open access 3D atlas such that each variance can be mapped to a current atlas. They also tried to develop a kind of a neuronal circuit in the mouse brain. So the mouse brain is kind of a easygoing, better than human, easy to handle with our data compute capabilities. So we really look at mouse brain for that aspect. So, and also we have, they are also looking at regions for specific cell-specific targeting, like different kinds of proteins that have been we can actually see some kind of retrograde as well as anterograde tracing, so that we can really look into that. And also we can also try to move into kind of human or non-human primate brain samples, like the previous lecture he was talking of the macaque brain or the marmoset brain. We do have we also do have kind of a part in the marmoset brain, which is kind of currently set up at Pittsburgh. So, but that is not that is kind of a different scenario. That is not part of this NIH initiative. So I'll talk of only mouse brains here. So also you can look at one of the Cell Press reviews just in October 2019, where you can see there is like they have actually looked into kind of the different opportunities and challenges present in while we look into machine learning and brain imaging together. For the computer scientists, the EURIPS is kind of a very prestigious conference, ISTAR conference in the field of machine learning, computer vision, and all those things. And you can really look that this year they had a separate workshop, which I think it was on December 14th, and you can look into that where it stands, like where the neuroscience and the machine learning are looking at together. This kind of what we are really seeing now. Has been a surge of people looking into neuroscience as well as machine learning together. So This is the next one is kind of Journal of Neuroscience from the Society for Neuroscience, the largest meet for the neuroscience groups all over the world. So they are also looking at machine learning where in neuroscience. So you can actually look into these kind of websites and these kind of journals to really get a view of how the machine learning is doing in this recent scenario. So this is just a very short overview. Like you can just really go to these, and you can also find links to different kinds of journals, which also links this machine learning as well as the neuroscience together. So just for the motivation, let me go a bit into technicalities here. So brain dynamics, or the different kinds of processing in the brain with the neurons, the axons, dendrites, when we actually inject some kind of reagents, the proteins into the brain, they require as well as kind of signal processing for both point and continuous data. They actually require kind of geometry. So what happens is we cannot, to the current scenario, we really look at slices of brains and image them. So we do need kind of geometrical techniques like diffeomorphic techniques where we need to actually map those images or the slices of the brain, whether coronal or sagittal. So we just need to stack those and map it to a reference atlas. There are several, 2 or 3 reference atlas available, mostly from, there are CCF 1, 2, 3, there are a few others. So you can just look at those atlas. So there are some diffeomorphic methods which map each of those mouse brains into this atlas. And also we need to look at the topological aspect, where we need to characterize the different kinds of structures in the brain. The structures of the neurites, which may be looking like trees within the brain. As we'll go forward, I'll actually get you through one of the slices of the brain where we imaged, and you can just look at how it looks like. The modern machine learning techniques are mostly data-driven, which is of course scarce in our case, and it mostly relies on nearest neighbor pattern matching, and also it requires a large annotated data volumes, which kind of really is a bottleneck. We are trying to ramp it up to be matching with other communities which are really doing good in the field of medical imaging. So mostly these techniques are very fragile to the different minor change in the data distributions. This is quite common in the field of machine learning, and so here we try to kind of combine the deepness with topological prior information. So what is geometry? What is topology? So when you look at geometry, what does it give? It gives you different kinds of things like what is a position, length, curvature, area, volume, whatever. But what does the topology means? It is mostly captured by connectivity. So for just look at these 2 images. When we see at kind of this cup as well as this donut. So when you look at the donut and the cup and the donut has the same kind of connectivity within each other. They have a single hole, they have the same kind of connectivity. So these 2 are actually the same when we talk of topology. But when we actually look into the geometry of them, there are different kind of position, their areas and volumes differ, their lengths and curvatures differ. So they are different in kind of geometry. So mostly for registration, we mostly use kind of geometrical techniques. But for the other parts, like looking at the neurites, different kinds of axons, dendrites, we need some topological priors to actually look into that information. So going forward, before going into what we really did with topological priors, let me give you what we'll really look into through today. These are the two kinds of brains which we will be looking. This is kind of a whole slide imaging technique which has been our mouse brain architecture project. On the left hand side, this is a fluorescent brain, and on the bottom uh, here, these are actually the cell bodies or the soma. These are few of the what we call as the fasciculated axons, and these areas, I'll show you something after I finish this slide. So these areas where you see there are certain dots which are actually, these are the classification. These are the original images, these are the classifications. So when you look at here, this is kind of a neurite, and on this neurite at these points, we actually can see some kinds of sudden change in variations, certain small dots. We call them these boutons. These are the synaptic swellings. And this is kind of a 2-photon image, which is kind of, you can tell, and this is a color image, and you can tell that this is kind of a serial 2-photon imaging, which is not a color, it's a grayscale imaging. So our brains are actually sliced at 0.46 micron spacing, and they are actually having a alternating Nissl and fluoro sections. But this is the brain from Josh Wang's lab, one of our collaborators in Cold Spring Harbor, which is actually around 1 micron spacing. So they are actually doing a 2-photon imaging, so we can see only grayscale images there. These are few of the classifications which we mostly look at the first stage of our of the algorithm, where we have this kind of two sites, for two kinds of tracing, the anterograde and retrograde tracing at the injection site and the projection site, and we do have some kinds of noise, what we call as artifacts. So in the injection site, you will have several soma, the cell bodies, at the axons, the dendrites, as well as these synaptic swellings. At the projection sites, you will have these axons, which will be further classified into fasciculated axons, axon arbors, as well as the swellings. In the retrograde tracings, you will mostly have these axons, and within the axons, we have all those, the fasciculated axon arbor and the synaptic swellings. In the projection site, we will have these dendrites, dendritic fibers, as well as the synaptic swelling, as well as we will have somas there. So this is kind of a generalized technique, but we don't say that we will not have somas. We'll have some sparse somas even in the projection site, which overspills from the injection area. So this is kind of an overview. Let me see if I can show you this. Just if I can get to show you. Does this go? Okay. Anyways, at the end of the talk, I'll show you some of these directly on the portal, where you can see all those images. So as we go forward, firstly, we need to look at few of the things. There has been a surge of machine learning, deep learning algorithms after the UNet. So UNet came as the winner of the ISBI challenge, where it came out as kind of a segmentation technique for the neural structures for several electron microscopic stacks. So this they actually won two of the challenges, and this is the cell tracking challenge as well as the between radiography at ISBI 2015. So these are few of the results which you look at. This is kind of a cell bodies which gets segmented out, and the yellow border is actually the ground truth. These are actually copied from their website, and this is also the kind of segmentation which we see, and the cyan is the segmented map of the UNet, and the yellow border actually is the ground truth. So the UNet is actually a kind of It uses a kind of a residual connections, the feedback connections. So based on this, there is another algorithm which we also incorporated. We try to incorporate into our technique. So they have kind of they call this a ALBU. It was kind of the first. It was the winner in the SpaceNet challenge for road detectors. So when you look at this road detections, you can see that this also looks like something like a tree structure. So when we look at these tree structures, we thought we can incorporate this. So this had a ResNet 34 as the encoder part and the unit-like decoder. Also, they use the skip connections and the dice coefficient with the binary cross entropy loss as the loss functions. For this, actually what they did was they had the whole data set. So they augmented the data set into 4 folds. So did a kind of a cross-fold validation, you can say. So they used the cross-fold validation for this. They used 4 folds together, and these of the 4 folds are mostly calculated as flips, horizontal and vertical flips of the images to counter with the different kind of pose variations in the image or the rotations in the image. So they actually reached kind of the first position with kind of 95% accuracy in road detection of 4 very populated areas. So what is our data and what are the challenges we are looking at? So when you look at this, this is a mouse brain. So and when we have a kind of injection here. So at the injection, we can see due to the injection at that point, the protein or the different kinds of neurons have been activated at different parts of the brain. So we need to form this connectivity so that we can find the source and target. This is kind of an anterograde tracing. So, and also we can see while imaging and we do get some non-uniform distribution, although this is kind of a connected neuron, but due to the change of variation in the intensity, we can see that this is not actually kind of being caught by the different kind of deep learning algorithms. This is just an example. There are several more examples in real life, so we can really look at those. So to handle those, we need to actually look into some kind of topological prior. For the topological prior, we looked at something called Discrete Morse. This is kind of a collaborative work with the Ohio State University under Professor Yusu Wang. And her 2 students, PhD students, Ding Kang and Lucas, have been working with us. So when we have actually kind of the tracer injected image tags, we convert them to a density field and construct a kind of a Morse-based graph, and we do simplify based on some thresholds, and mostly I actually put this figure in 3D so that you can understand, because if I put this in 2D it will be really tough. So I'll be switching on to 2D after this. So what we look at is like image to a kind of a scalar density field. So you look at this. So each of these, we construct a kind of a terrain with mountains and ridges, and we put these different kind of intensity values on this on the terrains, and this is the first table manifold which we actually try to extract. That means mostly in easier words, we try to extract the ridges from these mountains, and when we and each of these neurons we consider as mountain ridges as the neurons. So based on this, we can extract the different the faint connectivities on the graph, and this can actually handle different kinds of intensities in the image, even the higher intensities where the injections are, and away from the injections where it is at very low intensities. So this is what we actually try to include as a topological prior. And so this is kind of a mathematical interference of that. I don't want to go deep into it, so you can just interpret that the first stable manifold, that is from the d minus, means from the d to the d minus 1 saddles, if we take the gradient, is the ridges of the mountains, and which has been classified as the neurons. So for further discussion on discrete Morse theory, actually I listed down few papers, so you can look at those. The oldest paper, if you want, that is the classic Morse theory, if you really want to know what the Morse theory is, in 1963 paper, and the discrete Morse on Foreman. This is the basis of the work done in 1998, and the graph reconstruction, which I showed mostly on those mountain ridges, has been proposed in 2018 by Yusu's group. So moving forward, so why do we need this discrete Morse-based neuronal skeletonization? So when we look at those images, we can really see that these images are having different kind of intensity variations throughout the section, throughout the coronal stage. So if we are at the injection region, the intensity varies a lot. The intensities are very high, and when you look at those, very away from the injection region, the intensity falls considerably. So when the intensity falls considerably, we really need to look at a method which is robust to this kind of intensity variations. And also these images, which another better advantage of this algorithm is, mostly these medical images are captured in 16-bit. So this, but all these new deep net algorithms, the machine learning algorithms, the image processing algorithms mostly work on the 8-bit data. So this is also being robust to a 16-bit data. We can handle those. So this is a kind of mathematical model mostly, so it can handle any amount of data you give it to him, whether it's 16-bit or 8-bit or 12-bit, whatever it is. And it's very conceptually clean and easy to implement. So this is the topological prior we include into our algorithm. And this is the algorithm which we have been using to actually classify out the different kinds of neurites. So when you look at this, we have a Siamese neural architecture. The top part, we call it the topological pathway. Consider those contoury layers as kind of normal convolution layers with a ReLU activation. So we have that similar Siamese convolution networks both sides of the channels. We call the lower one as the neuronal pathway. So as we have seen in our images, the brain architecture project has kind of a 3-channel. Although it has a 3-channel, mostly we deal with kind of 2 kind of tracers. One is in the green channel and another is in the red channel. So we do all of this channel wise, mostly because this information interferes with each other, so we want to minimize that interference, so we do it channel wise. And also for the computational complexity, if we try to do it at 3-dimensional, the computational complexity really goes up higher, and also the turnaround time becomes much higher. So we do it on a single channel, and we pass it through that DM algorithm, the topological algorithm I was discussing, and the ALBO architecture, and then we actually stack them into kind of a concatenated stack and pass it to a common pathway to get to the output. So what does this tool really look like? The topological prior is mostly incorporated to actually mimic the brain's ability to fill up gaps where it sometimes when we see the gaps we can fill it up, even there is some faint signals, very faint signals, so we try to use the topological prior to mimic that. And also when we look at individual tracers, so the individual tracers at individual channels, so red as the green channel we are talking about. So the connectivity has to be designed for individual tracer. The reagent that has been put through it. So the design of connectivity will be actually affected by the individual tracer. So we generally focus on individual color for the design of connectivity. This is one of the explanations why we do it channel wise, other than the computational complexity I was talking about. And also this is kind of the flow diagram which we incorporate. So when you look at the images of what we are looking at, this is under 0.4 micron and we are having a very high dimensional image. So the images which we see on the web or which we actually have it registered to an atlas, so we have them at around 24K cross 24K pixels. So that is not what a normal machine learning algorithm or a computer can handle at this stage. So we have to really tile them up, then put them to the and put each of these tiles, their individual channels to the neuronal and the topological pathway, and then put it to the common pathway of DM++, such that we get a process mask. We call there different kinds of collection of the neurons, dendrites, as well as the other neurites, axons and the neurites, and whichever unclassified between them, the all neurites will be as processes. We output them as a process mask. And as a byproduct of the DM, so you saw that DM actually has kind of mountain and ridges. So when we have those mountain and ridges, we can easily find out based on the intensity, the peak of the mountains. So based on the peak of the mountains, we can really find out those synaptic swellings. So there is kind of a Bouton detector which will also be incorporated as a byproduct of this pipeline. To talk of what we face as the limitations, the hardware limitations. So of this network, specifically of this network, is like when we try to incorporate them, the major bottleneck is like ALBO is a 4-fold network. It has 4 models trained together. So that actually will kill up 90% of your GPU resources. If you are working on a very high-end GPU like 2080, 12 GB GPU, this will eat up to 90% of your resources. The next part is the Morse code actually is like it will calculate each and every path within the image. Although I was working with a kind of a 512 cross 512 image, I can tell you that it can compute up to 5,000 paths. So each of these paths, even if you parallelize on a CPU, how much can you parallelize? The number of threads. So if it will be very highly CPU expensive process, the Morse code. Although it's like a 16-bit data, and also there is since this is a SAM-edge network, so this will be highly expensive for a GPU as always for all the deep learning techniques. So there is one more drawback in this method. So what we are looking at is like 512 cross 512 tiles. So although this has been sliced, so we already have lost the 3D correspondence of the different neurites. But again, tiling them down to a smaller tiles, we lose even the 2D spatial correspondence. So this is kind of a fast pass process. So we are lagging behind due to the deficiency in the hardware field. I think on the hardware day you heard that how they are trying to build up on the different kinds of GPUs, different kinds of special purpose machine learning units such that they can really handle these. I don't know how long it will take to handle, but currently we are losing these. So uh, despite this, we do have some good results, and although since these processes are like kind of color channels, so we have difference in intensity values while we capture the images within two, means there are deformities in the brain cells. All these factors taken into consideration, we try to reach some kind of good accuracy in the field. And also I just mention a few major drawbacks there, which has been also kind of a unavailability of training data, which we are trying to build up. There has been this CCBR workshop, which we are currently a part of. The team currently has 5 people who are really sitting down and annotating under Dr. Jay. So we are team and annotating data for us such that we can really get into machine learning, and we do have data. But the machine learning for annotating a brain, something around 300 sections if we talk of only the fluoro sections, and 24K cross 24K tile manually will really be a kind of a bottleneck in the whole pipeline. So as we go forward, there is also a kind of speed up which we have thought of and we have implemented into our pipeline. So these are few other process detection results. So this is the single 2-photon imaging from the Josh Wang's lab. So we can see that this is the original, what happened, where is the mouse? This is the original image. So look at this. There is a faint connection in the original image, which even the human brain cannot interpret. So this is first pass of annotation. So human looking at it will be really bored and his eyes will be strained. So he will really not be able to delineate that very faint process. And once we go forward, this is the discrete Morse topological framework. It actually captures that from this image. And this is the neuronal pathway which actually is unable to capture this, and when we combine this with this, we get into our network which actually captures this. And this is just a kind of a evaluation when we actually try to evaluate our annotation with the ground truth annotation. So these things which we actually see can be again sent back to the annotators for corrections, and such that we get a good training data, we get a kind of better annotation back, and all our results which we see further will be much refined. And so this is not the end of the process. We have a huge pipeline going beyond this process detection. So once we have the human annotators sitting down and correcting these, or correcting themselves, or correcting the the automatic output, so this will be the first pass. So once we get kind of a good confidence that we don't need those kind of annotation, there can be these human annotators will also always be a part of our group, which our pipeline, where they will actually QC whether they will quality check whether the results are good or not. They will not annotate that time. So this is the future vision which we are looking at, but currently we have these annotators looking at this, just correcting our results, correcting their annotations, like this they are going forward. So sorry, I think this is the kind of the mouse brain architecture project where we have, so I told you there are two kinds of tracers, the green tracer and the red tracer. So this is actually very much enhanced for your visualization. So if this is a 16-bit image, you will not be able to see a single thing inside it. So The annotators annotated it. These are the green tracers they annotated. These are the red tracers they annotated. And in the discrete Morse, we have these green and red tracers. So everything is performed channel-wise. So this is the output of the neuronal pathway. And when we look at the DM++, the combined network, we can see that this connectivity is resumed by the discrete Morse and the neuronal pathway. And we have this evaluation. So actually, when you look at this kind of things, kind of this annotation, it's always there. There is always a pathway you can look at this. And this is actually taken as a false positive. Means I detected it when human annotator couldn't find it, but this is not a false positive here. So actually, so there is a need of improvement of annotation. So we need a feedback network from both the annotators and the algorithmic developers, such that we can really get this pipeline working and reaches to a stage that it becomes fully automatic. So this is kind of a process detection result overlaid on a human brain, and these are the these are the different kinds of, on the right hand side there are the different kinds of compartments of the brain, the different sections of the brain, and the green overlays shows that how the injections travel through the different sections of the brain. Going forward, so this is one of the downstream step which we can see from our from the process detection pipeline. This is a 3D. So we have this process detection done on all the sections of the brain. So we can stack them up, and once we can stack them up, we can actually stack them up to see that it actually matches the whole 3D brain. So this is actually a 10 micron voxels stacked to a 10 micron voxel level. So this is this is kind of a 3D visualization of the whole injection in a STP brain. So this is the downstream process, and this is the feedback mechanism which I was talking about. So there is two persons, the annotator and the algo developer. The algo developer goes to process detection, as well as the input from the annotator will come to the process detection, the annotated tiles, and we compare with ground truth. We determine the false positives, and we revise the annotation. We see the false negative, we revise the algorithm. So this has to be a process which has to go a few times in the near future, such that it can reach kind of a very good accuracy. So this is an example from the serial 2 photon. This has been done in kind of Josh Wang's lab. We have these two annotators, and when the first annotation when they gave us, and we then trained our algorithm on those annotations and sent it back to them to see which is false positive, which is actually a false positive. We didn't look at it. We looked at it, okay, this is not a false positive. I think human annotators is always prone to human errors. So we said that these are not false positives. We must send it back to them. So actually, when we send it back to them, the false positives actually reduced by 10%. So, and we can see that we have kind of reached an accuracy of around 90%, which we're aiming at the first pass of our algorithm. So the next part which we will look at is like the different semantics of the brain. So I'll just focus on one part, the cell detections. So this is the injection site. This is a 16-bit image, and when we look at the injection site, we can see we can see a few of these cells. So when we see these few cells, these are detected by one of our algorithms. It's kind of very common in computer science or the machine learning field, the mask RCNN. So So why did we choose mask RCNN? So the first part was we are looking at faster RCNN for the bounding box over the cells. So we needed the cell centers, so we thought bounding box would be fine. But suddenly there is kind of a cell detection framework. There was a Kaggle challenge where we can see that there are people who actually did automatic nucleus segmentation using mask RCNN. So we jumped into that, and this is like, although mask RCNN is just like improvement over the faster RCNN, and we also needed all these because the faster RCNN, we cannot go to a very traditional time consuming technique because our data is huge. Think of 300 images, each of them 24, each of them some more than a gigapixel image, and we need to go through all of those. So for a single brain, and we have almost 3,000 brains already imaged. So we need to have something very fast, we need to have something very effective, and we will always have kind of a manual intervention in the middle because this is not kind of the first, this is the first try, so we will need those manual interventions, and this is what we are looking at. So we jumped into mask RCNN. This is kind of the very basic network. This is kind, okay, this is this, if you really look at these things, these are like the rabies infected marmoset brain cells. So we do get kind of output mask with different kinds of cells detected. So I was just talking of the cell detection, but we are really looking at all these kind of different different kinds of semantic segmentation within the image. So we are looking at the injection detection, which is kind of the determined by the injection somas, where we saw the first image for the cell detection. We have those somas, those are actually on the injection site. I took out the injection site just to have a view of it. Then we have the process detection as one of the results. So based on the process detection, we can also have these boutons, which we saw the synaptic swellings. Then we can have these axon arbors, the fascicular axons, the dendritic fibers, the injection somas, and then post them to the web portal. So the posting to the web portal and actually making this whole thing available was one of the BICCN objective, such that it becomes by 2025 they're aiming to have this on the, have all of these for the mouse kind of well determined and well performing to the best by 2025. So they actually want it to be public. It's a National Institute of Health initiative. So this is the evaluation technique we use because this is somewhat tailor-made, but yeah, we may look at something very generic, but we cannot really rely on these algorithms because we are losing spatial information, we are short of data. So we actually thought of this thing. So if we have a pixel, this is a kind of pixel classification technique. So if we have a pixel within a radius of 5 to be near to a ground truth, we consider that as a correct classification. So otherwise it's a false positive, or if we don't get one, it's a false negative. So we calculate the different precision recalls. So this is one kind of a very not a mature but kind of fast pass table for the different kinds of brains, the STP brains, the MBA brains, the BFI are the bright field imaging brains, where we actually kind of look at those, what I said, the marmoset brains with rabies infection was kind of bright field imaging. For the cell detection, we had some kinds of results. We do reach kind of 90% accuracy, but we are striving to reach farther than it. So semantic category segmentation is kindly lagging a lot behind. This is mostly because of the different kinds of artifacts, the imaging artifacts, as well as few cells have their own autofluorescence. Sometimes there is some kind of noise. some kind of dust particles. So everything comes into a physical process. This is kind of physical process then image. There is nothing. So we do really suffer from all those. This is mostly what kind of the pipeline which is working on the data analysis task in the in CSHL and on Mitra lab. So you can see this yellow blocks. These are the manual parts, and this is mostly done in IIT Madras. So the CCBA team under Jay is really working hard to actually get this thing. They are actually getting all this proofreading done. They are the kind of the integral part where we will actually get kind of very fresh and very refined data to be working on the steps behind this. Okay, this is kind of see this kind of cell detection. Although I am just not very crudely, this is kind of development from one of IIT Madras people. So he is actually a kind of running a healthcare system in our research park. So Mr. Kirtiram. So he is actually developing, this is kind of annotation tool which the annotation team is working on. So these are few of my team members which we are actually working on. Before ending this, let me just get you something. What? Okay. Oh no, sorry. So, this is a mouse brain, okay? So what are we looking at? These are few processes in the brain. So I told you there are two kinds of injections, tracer injections we are looking at. This is kind of the green injections, this is kind of the red injections. So let me first show you a few things, which are the different classes. These are the fasciculated axons. The fasciculated axons means this the axons which are actually got strangled together. So can you look at these small dots? These are the synaptic swellings, the botons which we try to detect. They always lie on axon or a dendrite. Although this is not an injection site, this is an anterograde injection, so we will not have a cell, but luckily we have one here. So when you look at these, we do have these results on the web. So once we try to superpose you can let me move this out. How to move this out? Yeah. You can see that there are several processes detected here and there on the brains. So these are the different neurites that have been detected out of those injections. So this kind of available on the web on our brainarchitecture.org. This is kind of a development website. Don't go this. Go to brainarchitecture.org, and you can really look at few of these brains already have their process detections. Also, there are a few brains which will have the cell detections on. Even this guy on the STP brain, you have the process detection mask on. So you can look at those. I think, yeah, that's all I can really talk about now.